This is Unit 5B, Part 2. We're discussing muscle and how it gets activated, skeletal muscle in this case. So, as I was saying in the first part, motor neurons that are part of the somatic nervous system, they innervate the skeletal muscle. So the axons in the nerves come down to the muscle, and then their endings split apart, and they come very, very close to these endings, come very close to individual muscle fibers. And at the end of those axons of the motor neuron are the endings, and they contain vesicles, membrane-bound structures, phospholipid membrane-bound organelles inside the endings, and those vesicles contain neurotransmitter, and the neurotransmitter of the somatic nervous system that innervates skeletal muscle is always acetylcholine, and it's always excitatory. So where that axonal ending comes close to the skeletal muscle fiber, that's called the neuromuscular junction, where the axon, the end of the axon of the motor neuron meets the skeletal muscle tissue. The area of the sarcolemma, the plasma membrane, in other words, of the skeletal muscle fiber that's immediately across from the endings of the axon, that's called the motor end plate. And on the motor end plate, you have acetylcholine receptors. So those axon endings, even though they're very, very close, they don't touch. There's a space there called the synaptic cleft. And this diagram shows that kind of structure, where you have here at the top the diagram showing the myelin coated axon of a motor neuron coming down and then innervating this skeletal muscle fiber. So, and this of course is just a short section showing here the skeletal muscle fiber can be much, much, much longer. You see let me change colors here. You see the multiple nuclei on the edge. You see the striated pattern, and you see the axon terminal that contains vesicles of neurotransmitter. What happens with activation, I'll go through it, but we'll also see it on the next few slides. What happens when an action potential comes down, an action potential impulse, in other words, is sent down the axon of the motor neuron, and it arrives at the axon terminal, then what happens is that calcium channels open and calcium flows in. When calcium flows in, what happens is that the vesicles, and they're shown here in green, these round green vesicles that contain the neurotransmitter acetylcholine, these synaptic vesicles, then with that influx of calcium, they're able to move to the presynaptic membrane of the axon and fuse with it and release their contents, acetylcholine. And the acetylcholine neurotransmitter goes into the synaptic cleft, the space between the skeletal muscle fiber and the axon at the neuromuscular junction. That space, the synaptic cleft, and it diffuses across and then can bind to acetylcholine receptors that are found on the motor end plate, the sarcolemma at the motor end plate. This area here, I'll mark it in, all of this area here is called the motor end plate. Okay? And this junction between the axon and the skeletal muscle fiber is called the neuromuscular junction. At a higher magnification shown here, what you see is the synaptic vesicle fusing with the membrane, the acetylcholine molecules diffusing across and binding to channels, in this case sodium channels and also potassium channel. And as you know, potassium is higher inside than outside, and sodium is higher outside than inside. So, well, the initial response, of course, is that sodium goes in. Some potassium can go out, and that tends to make the inside of the skeletal muscle fiber less negative, more positive. Okay, so you have openings of sodium, potassium channels. Sodium comes in, potassium can go out. The acetylcholine comes, when you have this kind of receptor-ligand interaction between the receptor and the molecule it binds to. Remember, all these receptors are very specific. So the acetylcholine receptor binding to acetylcholine is not permanent. The acetylcholine comes off, and then it actually gets split apart by an enzyme called acetylcholine esterase. That way, you don't have constant, continuous activation or excitation. So, as I was saying, the nerve impulse reaches the end of the axon. Voltage-regulated calcium channels open. Calcium flows in, causing the fusion of the vesicles with the membrane and releasing acetylcholine into the synaptic cleft. It diffuses across and binds to acetylcholine receptors, and that initiates an action potential or a depolarization on the skeletal muscle side. So if this is, for example, if this is the axon coming down and there's the axon ending, and if this is the skeletal muscle here, then what you have, of course, is the action potential coming down, activating calcium channels, and you get the release of acetylcholine into this space, and you'll get a depolarizing effect because of the effect on sodium and potassium channels on the motor end plate. The motor end plate is this area here of the skeletal muscle fiber sarcolemma. This change in membrane potential, if it's large enough, will trigger an action potential in the skeletal muscle fiber. As I said, the acetylcholine bound to the receptors comes off and gets destroyed by the enzyme acetylcholine esterase, and that prevents continuous muscle fiber activation and, in other words, contraction. It doesn't continually contract. It'll stop unless more signals come down the axon. So now you have a depolarizing event happening at the motor end plate on the sarcolemma. You get a reversal, and if, as I said, it's strong enough, then you'll get an action potential. The depolarizing effect will spread away from along the membrane, the sarcolemma membrane of the skeletal muscle fiber, away from that area, and it'll spread away. If it's large enough, you'll get a triggering of an action potential that spreads away from that area because of the opening of the chemically gated sodium-potassium channels, and sodium moving in and potassium going out. And you'll get depolarization of the muscle, skeletal muscle fiber. That local event is called the end plate potential. It can generate, if large enough, an action potential that spreads in all directions away from the neuromuscular junction. Here you see relative levels of calcium and potassium on the inside and outside of the skeletal muscle fiber, a section of skeletal muscle fiber where the potassium is higher inside than outside, as you can see, and sodium is higher outside than it is inside. Normally, the sarcolemma, plasma membrane of the skeletal muscle fiber, is impermeable to both. However, with the release of acetylcholine, you get an increase in sodium channels opening, so sodium enters, and you get depolarization. It goes to more positive because of that positive sodium accumulation, entry of positive sodium channels. And again, as it, just like pretty much similar to the idea of with neurons, if the stimulus is strong enough, an action potential is triggered, it's initiated. And so this area here would be the motor end plate, and sodium's moving in, and that spreads. So you get a spread to the adjacent patch, and that patch is has sodium channels which now open, and that depolarizes, and the action potential rapidly travels right along. You know, this whole process of each adjacent area becoming more positive with sodium channels opening, then it travels right along the length of the sarcolemma, moving away from the neuromuscular junction, the motor end plate. The action potential at this point is not stoppable. It's all or none always, and it will result in the contraction of the muscle. Now, right after that depolarizing event occurs with sodium channels opening, then the sodium channels close and potassium channels open. Now, potassium being higher inside the skeletal muscle fiber than higher inside than outside, then potassium will move out to the outside. It goes along the concentration gradient, and you get repolarization, and the normal resting potential of the neuromuscular junction is restored. It gets restored by the action of the sodium-potassium pump. So things go back to the way they were before. Now the idea here, however, is it gets more complicated because there is this excitation-contraction coupling event that has to occur. It's not just enough that the action potential spreads along the surface of the sarcolemma of the skeletal muscle fiber, moving away from the neuromuscular junction motor end plate, right? That action potential spreads, and not only spreads along the sarcolemma, but then it dives down the T-tubules. And if you recall, the T-tubules come down into the inside of the skeletal muscle fiber along the walls of the different myofibrils that are inside, and it'll trigger calcium release. That action potential going down the T-tubules triggers calcium release from the terminal cisternae of the sarcoplasmic reticulum. At this point, now is when you start getting, with that calcium release, internal calcium release inside the skeletal muscle fiber, you start getting activation so that of the myofilaments, the actin and myosin myofilaments, so that thick and thin myofilaments, so that you can get contraction. Calcium will bind to troponin, and causing the blocking action of tropomyosin to stop, and the actin binding sites on the actin binding sites get exposed. So what you have is, with exposure of that, you get myosin cross-bridging from the thick filaments. They attach, then they detach, things slide along, and the thick and thin filaments slide past each other. Okay, all of this is driven by ATP. Energy for it comes from ATP, and once the stimulus from the motor neuron is stopped, then the calcium stops being released, and calcium gets removed by being pumped back into the sarcoplasmic reticulum, into the terminal cisternae. The tropomyosin blockage of the troponin is restored, and the muscle fiber stops contracting, it relaxes. All of these events that I've just described are shown on this diagram. Here you have, with the arrival of a action potential down the axon, you have entry, remember, of calcium triggering the synaptic vesicles to bind to the presynaptic side of the membrane of the axon ending, releasing acetylcholine that drifts across, binding to acetylcholine receptors, causing sodium to move into the skeletal muscle fiber, into right at the site of the motor end plate. Sodium moves in, so you get a depolarization, and if you get enough, then an action potential spreads along the sarcolemma, away from the motor end plate area, and will go down the T-tubules. And the effect of that is that it will trigger release of calcium from the terminal cisternae of the endoplasmic reticulum. And what that does is it binds to troponin, and the troponin will change shape, removing the blocking action of the tropomyosin. And that allows the myosin head to attach. Remember the myosin heads that are at the end of each filament of the thick filaments that are made of myosin. So this head structure looks like the end of a golf club, kind of. It will bind to actin and pull the actin filament along. The release, okay, with binding you get that movement. So the movement is shown here in this direction. They effectively, the two are sliding past each other because of the action of that myosin. And then you get release of this myosin head, and that's what requires some energy. The release of the myosin head from the actin filament, the thin filament, requires energy. So it releases, it moves down like that, and then will again attach to a new site further along. So it now attaches maybe, further downfield. You might have an attachment site, and then again it ratchets along. It pulls the thin filament past the thick filament. This all stops when the action potentials stop coming down the nerve, and the calcium gets transported back into the sarcoplasmic reticulum, and the blockage of the actin binding sites is reestablished. Tropomyosin again starts to block those actin binding sites so that it can't bind to the troponin. So that's how things work in terms of excitation, contraction, coupling between neuron excitation and the contraction of the muscle. So this diagram is showing the interaction between the myosin thick filament and the actin thin filament, the two types of myofilaments. And here in this case you have the myosin cross bridge attached to the actin myofilament, and now with a following attachment you get pivoting of the head. You see that the head changes its angle, and it's attached here in number 1. It's attached to the actin thin filament, then it pivots, and that pivoting action is what moves the thin filament, slides it in that direction. So the thick and thin filaments are sliding past each other. That pivoting slides the two myofilaments past each other and causes the sarcomere to shorten in size, as I showed you in the last unit, Unit 4B, Part 2. And you get release of the myosin head from the thin filament when ATP comes in and binds to the myosin head. So ATP, an energy source, is required for release. With attachment, the cross-bridging detaches. The ATP gets split into ADP and phosphate, inorganic phosphate, and the head returns to a state that's ready to be activated to attach again. Okay, so this kind of movement of the two past each other, where if we do this like this, say that's the thin filament and this is the thick filament, you're getting the head attaching, and first it attaches vertically in a vertical direction, and then it pivots, changes its shape, and so that it's going that way. And that what that causes is it causes the two to move past each other. It causes the thin filament to slide in this direction. So the two are sliding past each other and causing a shortening or contraction of the sarcomere. And then when ATP again comes in and binds to the myosin head, then it can release. And the head goes back to a position where it's ready to attach again, given the right signal. And that's the end of Unit 5B, Part 2.